In this video, we will discuss about this one-step optimized CAR NK validation in the in vivo studies. In the previous video, we have understood this optimized one-step CAR NK methods validations in the in vitro studies. So here they did the similar studies that we have discussed in one of the figure of this paper. That is, they used in this case the Raji tumor cells. In the previous studies of the in vivo system, they used some different tumors. So what they did is they took the mice, irradiated it so that there will be less tumor rejections, and thereafter 2 days after this irradiations they inject this Raji tumor cells, and 2 days after that they inject their optimized CAR NK, and then they did the measurement at day 0, day 7 and day 14, and in this period they also inject this interleukin 2 to keep these NK cells in the activation state. And what they are able to, and they use this optimized one-step optimized CAR NK validations in 4 different cases, and they divide them as group 1, group 2, group 3, group 4. Group 1 is untransduced allogenic NK cells. Group 2 is transduced allogenic NK cells with only the CD19 expressions, CD19 CAR expressions. And group 3 is transduced CAR NK cells with CD19 expressions. At the same time, this HLA-ABC is been knocked down using the CAD-RNA 1, and this PD-L1 is overexpressed. And the group 4 is the allogenic CAR NK cells, along with these allogenic CAR NK cells with CD19 CAR expressions, and they also have the suppression of HLA-ABC and the overexpression of HLA-E through this SCE, which is the optimized sequence that they have developed, that we have discussed in one of our previous videos. And what they are able to see is that in the case of group 1 and group 2, they are able to see a rise in the tumors from the IVS imaging, but for the group 3 and group 4 they are able to see reductions in the tumors. So, and one more thing that I forgot to mention, the Raji cells that they induce or inject into the mice is luciferase conjugated. So they are able to see the luciferase signals if the tumors grow up. So that signals is down in the group 3 and group 4, but they are high in the group 1 and group 2. And when they quantified this thing, they are able to see that group 3 and group 4 are able to suppress the tumors progressions, and with group 3 is performing slightly better than group 4. And when they check the survival rate, they also find the similar results, that is group 3 survival rate is highest, and then group 4, however the group 1 and group 2 survival rates are the minimum. Thereafter they wanted to check, or they also checked the amount of this allogenic NK cells in different conditions at different time interval, and they are able to see that for the case of group 1, the amount of untransduced allogenic NK cells initially is around 80%, but it goes down to 10% after day 14. And for the group 2, they see the same things, that the untransduced allogenic NK cells goes down, and these things are true for group 3 and group 4 also. And the transduced NK cells also goes down in the group 2, but they increase in the group 3 and group 4, indicating that the group 2's transduced allogenic NK cells are also being rejected, whereas group 3 and group 4s are increased. That means they are able to evade the immunogenic response by the host immune cells or host immune system. And when they inject this optimized CAR at day 0, they inject it with this 50% of this PBMC cells, their respective donors' PBMC cells. And the panel E is the results for the blood samples collected from the blood, and panel F is the results from the sample collected from the tissues. And here we can see that the host NK cells dominate in the group 1 conditions and group 2 conditions, but they are minimum in group 3 and group 4, indicating that there is a continuous expansion of this allogenic NK cells with reductions in the HLA-ABC and overexpression of either PD-L1 or SCE. So they are able to and this could be the reasons for the suppressions of the tumors in group 3 and group 4. So thereafter they wanted to see what NK cells are which NK cells participated in they did the further studies to find out which NK cells participated towards the suppressions of the immune system, and they are able to see that and they checked it using the CD107A, which is the degranulation marker, and they are able to see that the degranulation markers is high in the group 2, 3 and 4 for both in the bloodstream and also in the spleen. So this indicates that the allogenic NK cells are the one that is fighting with the tumors, and the host NK cells levels remain same in almost all the conditions. So it's the allogenic NK cells that is responsible for this anti-tumor response. Thereafter they wanted to see the reason for since they considered that the reason for the rejection of the untransduced and the transduced only with CAR NK, the CD19 CAR NK cells could be in the host T cells response. Host T cells might have eliminated it. They validated these things by checking the activated T cells markers, that is CD69 markers, and they are able to see that in the case of group 1 and group 2 they are able to see more CD19 T cells compared to the group 3 and group 4, and the minimum amount of CD19 T cells are in the group 3, and indicating that this could be the reasons for the highest efficiencies in terms of the tumor suppressions, as there is less immunogenic response, indicating there are more amount of this CAR CD19 CAR NK cells with reduced HLA and overexpression of PD-L1s are there compared to the group 4. So this results also makes the logical sense why group 3 is behaving more superior than all other cases, although group 3 and group 4 are almost similar in terms of its tumor suppressions and survival rate. So I hope you understood how they validated their one-step optimized CAR, and this indicates that the optimized CAR system can be produced in a single step, and while maintaining the almost same efficacies as we have seen in the multi-step optimized CAR NK method productions. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.